Lipids are composed mainly of carbon, hydrogen, and oxygen. However, some lipids also contain nitrogen and phosphorus. Some lipids are hydrophobic. That means they don't mix really well with water, but they are soluble in a nonpolar organic solvent like chloroform, acetone, and others. Lipids are made from hydrocarbon monomers. When many attach together, they form a long hydrocarbon chain used for long-term energy storage, regulating signaling, insulation, and protection. Fatty acids consist of carboxyl, a group on one end of a carbon chain, and a methyl group on the other end. They are vital for the operation of all body systems. Fatty acid double bond can either be cis or trans. That helps create different types of fatty acids, and they are classified by the carbon chain length and degree of saturation. When the carbon chain length is shorter, it becomes more liquid. There are three types of lipids that are important biologically: fats, phospholipids, and steroids. Fat is constructed from two kinds of molecules, glycerol and fatty acids. Glycerol is an alcohol. Each of its three carbons have a hydroxyl group, and the fatty acid is a long carbon skeleton. Unsaturated fat has one or more double bonds. The kinks where the cis double bonds are located doesn't allow the molecules from packing together close enough to solidify at the room temperature, such as olive oil. The saturated fats lack double bonds, and their flexibility allows the fat molecules to pack closely together, and it makes a solid at the room temperature, like butter. Phospholipids are diglycerides. They have two fatty acid molecules attached to a glycerol backbone, and the third acid has a phosphate group. This structure is what makes water soluble. The phospholipids bilayer is when the phospholipids are added to the water. They self-assemble into a double-layer sheet. They provide protection for the cell membrane, controlling what goes in and out. Esterols have a different structure from the other one I just talked about. They are complex molecules. They have 4 fused rings. They consist of interlinked rings of carbon atoms, with side chains of carbon, hydrogen, and oxygen attached.
So proteins are found in our food, they need for muscles, and they've got several functions to do in our body. And they are also the basic components of enzymes, hormones, antibodies, ribosomes, and many more materials. Proteins are multipurpose molecules. They are also involved in everything. Proteins, in order to function, they need ideal environment, and they've got several functions to do, as mentioned in the slide. The process of making proteins is called protein synthesis, and the proteins are synthesized in the body through a process called translation. So proteins are polymer molecules which are large and complex and have got 3D shapes. Polymers are molecules made from a large number of monomers joined together. In this case, the monomers are the amino acids. Amino acids are monomer of proteins which are made of central carbon, amino group, carboxyl group, and R group. There are 20 different amino acids in our body, and human can synthesize some of these. The R group is a variable group which is different for each amino acid. There are different side chains attached to a carbon, and this make the amino acid different from another. There are 2 different R groups, hydrophobic, which is water-hating, such as alanine, and hydrophilic, which is water-living, such as creatine. 2 amino acids joined together form dipeptide, 3 amino acids form tripeptide, and less than 20 form oligopeptide, and more than that form polypeptide. 2 amino acids are joined together by a peptide bond to form a dipeptide bond. This occurs when a molecule of water is released from the amino acids. This is called the condensation reaction. To form a polypeptide, series of condensation reactions are occurred. Vice versa, to split dipeptide into 2 amino acids, a hydrolysis reaction is occurred in which water H2O is added to break down the peptide bond to make 2 amino acids. For polypeptide, more hydrolysis reactions are occurred to have amino acids. Proteins' functions depend on the structure of proteins, and we have got 4 different structures. In the primary structure, the sequence of amino acid in the chain is linked by peptide bonds, as explained earlier by condensation reactions. And the polypeptide chains have directions to follow, and these directions are given by genes, which are made of DNA, and they also determine the order and the number of amino acids. The secondary structure is known as local folding, in which there are interaction between amino acids, and the folding is done with the help of hydrogen bonds, which are weak bonds, and the sequence of amino acids can be folded either in alpha helix or beta pleated sheet. When alpha helix or beta pleated sheet twist and fold around itself, a 3D shape is formed, and it's held in place by interactions between R groups and different amino acids. The interactions are disulfide bonds, hydrogen bonds, hydrophobic bonds, and ionic bonds. In the quaternary structure, more than one polypeptide chain is bonded together in order to function. All these structure helps the protein to be formed and shaped correctly in order to be functional.
Carbohydrates have many functions, such as storing energy in our cells, building macromolecules, and they are also the primary source of energy for our bodies. Carbohydrates also promote digestive health. For example, after a meal, our digestive enzymes break down the multi-sugars in the food, which are called polysaccharides. These then get turned into single sugars, which are their monosaccharide forms. Monomers combine together through polymerization to make polymers. Polymerization is the process where lots of monomers come together to form a large molecule, which is also known as polymers. This is shown on the bottom right diagram. Glucose, fructose, and galactose are the three main monosaccharides that make up carbohydrates. As said in the previous slide, there are three main monosaccharides. Glucose is used in cellular respiration and is very important for energy usage in our bodies. Fructose is found in an abundance of sugars, for example in fruits. Galactose is found in milk and has the same chemical structure as glucose, however the only difference is the position of one hydroxyl in carbon 4. The hydroxyl in glucose is at the bottom, whereas in galactose it's above, and this is shown in the diagram below. Glycosidic bonds combine two monosaccharides together, which make a disaccharide. There are three disaccharides. These are maltose, sucrose, and lactose. The bonds are formed by a condensation reaction that occurs when a water molecule is lost. The reverse reaction of this is called hydrolysis, which can split this bond by using a water molecule, and it also releases energy. Monosaccharide molecules form together and make long polysaccharide chains, which are also insoluble storage molecules. The three main polysaccharides are starch, glycogen, and cellulose. Starch has two forms, amylose and amylopectin, which are two different forms of glucose. Amylose enzymes contain lots of glucose molecules that have a spring-like shape. These molecules can only work on the ends, whereas amylopectin enzymes have glucose branches coming off them in different directions, making them much faster at breaking down. There are two common forms of glucose, alpha and beta glucose. Alpha is the building block for foods such as whole grain and potatoes, etc. They are not stable and can also be separated easily. Beta is used to create cellulose, also known as fiber, which is really important for our digestive system in order to function efficiently. The molecules in beta glucose are compressed together, making them stable and unbreakable. The only difference between alpha and beta glucose is the position of one of the hydroxide groups. When two specific monosaccharides join together, they can make certain disaccharides, as shown in the diagram. This reaction is called dehydration synthesis, which involves the loss of a water molecule and a covalent bond formation. They are formed by a glycosidic bond, which can either be alpha or beta glucose molecules. This all depends on the chemical structure. Dehydration synthesis is very important in order to make larger biomolecules.
Enzymes are globular proteins that are encoded by our DNA, and the unique folding of polypeptide chains is what creates the unique shape of an enzyme. The active site is a small section on the enzyme which the substrate can bind to. Once the substrate has bound to the enzyme, it forms an enzyme-substrate complex. Enzymes can be found inside of our cells as well as outside. Enzymes catalyze all biological reactions that occur in the body, for example, respiration and digestion. They're all split into 6 categories, which can be seen on the slide. Activation energy is the minimum amount of energy required to start a reaction. So for more successful collisions between molecules, we need an increase of energy to be used as activation energy. This is usually heat. However, many molecules have an optimum temperature, and an increase will lead to a disruption in temperature. For example, cell membranes, which are mostly made of lipids, will become too fluid in high temperatures. Instead of an increase in temperature, enzymes are used as catalysts, and they decrease activation energy by breaking or building substrates on a chemical level, as well as applying physical tension to break bonds. There are two methods which explain how enzymes work, the first being lock and key and the second being induced fit. Lock and key suggests that an enzyme is like a lock and the substrate is like a key, and they fit together perfectly. However, induced fit suggests that the enzyme is more like a glove and the substrate is more like hand. So once exposed to the substrate, the active site will induce a fit around it. The widely accepted and used model for how enzymes work is the induced fit because it clearly explains how catalysis takes place. However, there are advantages to the lock and key method. For example, the need for specificity in order for enzyme-substrate complexes to form. There are many factors that can affect enzyme activity, one being enzyme concentration. An increase in enzyme concentration will lead to an increase of the rate of reaction because there are more active sites available for substrates to bind to. However, eventually it will stop having an effect when enzyme concentration is no longer a limiting factor. Initially, a temperature increase will increase the rate of reaction. However, once it's high enough, the bonds that hold an enzyme together, for example, the hydrogen bonds, will become weaker and eventually break, changing the shape of the active site. Therefore, less reactions will take place. So an optimum temperature must be kept for maximum rate of reaction to occur. There are two kinds of inhibitors that affect enzyme activity. The first being competitive, and it works by fitting into the active site and blocking the substrate from binding to it. However, these are usually non-permanent and will be released by the enzyme and can be solved by increasing substrate concentration. The second is a non-competitive inhibitor, which works by inhibiting the enzyme product complex from forming. It binds to an allosteric site in the enzyme and changes the tertiary structure. These are usually irreversible, however, there are some non-permanent inhibitors.